Thank you, Cal, and thank you to OIS for again having us here today. Good afternoon, everybody. I just quickly passed our forward-looking statement. We are a public company based in New York City. And the goal of IVERIC Bio is to become a leader in developing transformative gene therapies for treatments for patients with orphan inherited retinal diseases. We've built a portfolio of 6 programs, and it's based on 3 criteria and principles. One is compelling science and potential best-in-class assets. The second is unmet medical need. The third is significant commercial opportunity. We're a relatively new company in the gene therapy area, so our focus on execution is very important. I'm going to talk about 2 of our lead programs in a moment, and they're on track for IND-enabling studies. We have CMC in place. We also would like to see these programs in the clinic later next year and the year after. People are very important to get this done. We have deep retinal expertise, which will be critical to bring these programs into the clinic, and we continue to build our team internally with gene therapy expertise. Our business development continues. I'd like to add to this portfolio over time, and our financial position is strong. As of June 30th, we had $107 million in cash, and importantly, we have runway through the end of 2021. This is our portfolio as it stands today. Our 2 lead programs, IC100 in RHO ADRP using AAV5, is planned to enter the clinic in the second half of 2020. Our second program, IC200 for BEST-related IRDs using AAV2 into the clinic in the first half of 2021. We have 3 mini-gene programs, which I'm happy to talk about in a moment. We're very excited about those, and also a program in AAV gene delivery technology. If we're fortunate enough to bring these products and solutions to patients, although this is an orphan disease category, there are a lot of patients impacted by this. On this chart here, we show prevalence. This is prevalence in the United States and the big EU5. Obviously, these numbers dramatically increase as you look at global prevalence of these diseases. But at the bottom, LCA10, which is one of our mini-gene programs with 3,000 to 4,000 up to Stargardt, which is considered a large orphan at 62,000 to 77,000 patients. We wouldn't be able to get here today without our scientific collaborators. We're happy to be working with the University of Pennsylvania and University of Florida on our 2 lead programs, IC100 and IC200, and with the University of Massachusetts Medical School and the Jorge Gene Therapy Center on our mini-gene programs. A little bit about our manufacturing strategy, that is to invest early to minimize process changes. This is one of the big hurdles in gene therapy today. We're happy to be working with Catalent's Paragon Gene Therapy Group, which has world-class capabilities and facilities. They're currently helping us right now in our process development work, and will manufacture our 2 lead program products, IC100 and 200. And also working with Devron supplying plasmids to the company. Just a couple words about each of our programs. Lead program IC100, RHO ADRP. We believe it to be potentially best in class. No clinical stage gene therapy competition at this point. Our strategy is a mutation-independent strategy that looks to knock down and replace with a single AAV vector endogenous mutant toxic rhodopsin protein and replace that with a healthy rhodopsin protein. I'm going to show you some images on proof of concept in a moment in 2 animal models, both canine, large animal models, and mice. The canine is a naturally occurring canine disease model. We've seen long-term preservation out to 9+ months. Believe we have a clear path to IND on this program. We've completed pre-IND FDA meetings, ongoing IND-enabling activities, and importantly, natural history studies. As I mentioned, manufacturing in place and also plan to be in the clinic next year. Some very important images from the canine proof of concept study. On the top, you see anatomy. Left side treated with a white bar showing outer retina rescued. To the right side where there's no rescue, and then also on function, green being the treated, red being the control. Again, good results here as well. Our second program, IC200, again, believing this is a first-in-class and best-in-class asset. No clinical stage competition at this point in time. Proof of concept here has also been demonstrated in a naturally occurring autosomal recessive BES1 canine model. 3 different mutations, and here, long-term preservation of retinal structure out to 4 years. I'm going to show you an image in a moment. Here also, clear path to IND, completed IND FDA meetings, natural history studies going on, manufacturing in place, and objective to get this in the clinic in the first half of 2021. Some of the images from the canine study, quite impressive. If you look at the upper left-hand corner, the control eye, and look at the red arrow below, a small micro-detachment at 103 weeks with no treatment. That micro-detachment continues to grow. And to the right, the treated eye with an AAV therapy, a human transgene. You could see at the beginning of the micro-detachment and at 103 weeks, full resolution, so quite impressive. Our next area are the mini-gene programs with the University of Massachusetts. We believe AAV vectors are an optimal way for ocular gene therapy. AAV vectors are safe and effective. We see that even with Luxturna, the approved product on the market. There is a limitation, and limitation is size. Working with UMass and the expertise they have there, we're working to take large functional proteins, re-engineer these, and put these re-engineered smaller functional proteins into existing AAV vectors, as you could see with the cartoon on the bottom. 3 programs here. First program in LCA10, that's our lead program, Stargardt's, and also USH syndrome, type 2A and USH 2A. Our lead program, LCA10, we've seen some very encouraging work here in terms of proof of concept in animal models with retinal anatomy and function being preserved and rescued. And that was the basis for us to sign a license with UMass in July of 2017. Our second program, which is a little earlier than that, but exciting work going on there, looking for a lead target. This work continues, and we should have some results of research in 2020. And a program that we just brought on, USH2A, which again is a large inherited orphan retinal disease, but also a very large protein, as you can see here, about 15 kb. Our objective here is to develop a mutation-independent mini-gene delivered with an AAV vector. Early work here, more to report as we move forward. We're also happy to be working with Dr. Guangping Gao at the University of Massachusetts on vector technology. We have some pilot studies ongoing currently and expect some histology before the end of the year. So we've come a long way in a very short period of time, and in summary, our 3 lead programs are on track, the first 2 to be in the clinic hopefully next year and the first half of the year after. Very excited about the work on our mini-gene for LCA10, in which we just signed a license agreement, added 2 more programs to this. And as I mentioned, you don't do this without great people, and we continue to expand our internal network, our external network, as well as manufacturing, which I talked about before. So I'd like to thank everybody for listening today. Thank you.